So, as we learned from the earlier lectures, genes control the inheritance of traits and are located on chromosomes. So, in this video, we're going to learn about exactly how DNA stores this genetic information and how this information gets turned into observable phenotypes in organisms. So, we know about Mendel's heritable factors, and Morgan's genes on chromosomes are made of DNA. And DNA is the biomolecule that carries all of the genetic information that an organism needs in order to operate in life. And collectively, it defines what an organism can do and what it would be. So, because of that, it's very important for a cell to duplicate its DNA accurately and pass it to the next generation during cell division so that the daughter cell or the offspring will be able to carry all the genetic instructions the parents so they can do the same thing. So E. coli would make more E. coli, and humans can give birth to more humans, but not necessarily other species, right? Okay, so if you remember, DNA are made up of nucleotides, right? So each nucleotide here has a pentose sugar. So here's our sugar, and it's attached to a phosphate group. You see the P here, phosphate group. And then it's attached to one of 4 nitrogenous bases. So you see our N's here for nitrogen, right? And you'll see that the structure of these 4 are different. And that's because we have adenine, thymine, cytosine, or guanine. So we just call them A, T, C, G. And earlier this semester, we learned that DNA is a macromolecule that's made up from many nucleotides bonded together through covalent bonds between the phosphate group of one nucleotide to the pentose sugar of the other nucleotide. And these form a long polymer chain called a polynucleotide. So all of these together would be a polynucleotide. Okay. So the way DNA store genetic information is by using these 4 nitrogenous bases. So just like English is made from 26 letters, and by placing these letters in different linear orders, they become meaningful words and sentences. So the genetic language is written in a linear arrangement of these 4 DNA nucleotides. So this means if we see A, T, T, C, G, C, G, G, and then we see A, T, C, G, C, G, T, T, A, those would convey 2 different instructions to the cell. And since genetic information is stored in the linear sequence of the nucleotide bases, right, technically one polynucleotide molecule is sufficient to do the job. However, each DNA is composed of 2 strands of these linear polynucleotides with alternating phosphate to pentose sugar backbones on the outside and the nitrogenous bases here in the middle. So we'll see in a minute why this structure is crucial. So for each DNA strand, its 2 ends are very different. So one end has the phosphate group. We'll see here our phosphate group here that's attached to a number 5 carbon pentose sugar, right? And this is called our 5' end. So we have our 5' end, while the other end has a hydroxide group attached to a 3 carbon pentose sugar, and that is called our 3' end. Okay, so it always runs 5 to 3 here. And then the 2 strands run in opposite directions. So you see our nitrogenous bases here in the middle. A and T always go together, and C and G always go together. Okay, so these 2, this is one big strand over here, one strand of DNA, and then we have one over here that are going to go into that double helix, right? And you'll see we have the 3' end up here that corresponds to our 5', and then we end with the 5' down here that corresponds to the 3'. right? So we call these strands anti-parallel to each other. So you'll see anti-parallel down here. Okay. So the two strands are held together by hydrogen bonds here that are formed between these two nitrogenous bases. And these dashed lines are what symbolize those hydrogen bonds. Remember, hydrogen bonds are very easy to break apart. And just like all other biomolecules, DNA have a very specific shape. Again, remember this double helix. So our two polynucleotide strands twist themselves to form this very specific coil structure, our double helix that we see here, right? And the double helix structure of DNA molecule can only be formed when all the bases between the two strands are paired in a specific way. So again, adenine always goes with thymine, A and T, and they'll form two hydrogen bonds between them, while as cytosine and guanine always go together and they form three hydrogen bonds between them. So A to T 2, C to G 3. Okay. And only when the specific A, T, C, and G pairing occurs, all of those nitrogenous bases can be positioned into a fixed angle relative to that phosphate sugar backbone, and it allows this formation of the double helix structure. If there is a mismatch that deviates from the specific ATCG pairing, say one strand A is paired with C on the other strand, the bases would need to rotate in order to form hydrogen bonds between them, and this would end up causing a distortion in the double helical structure of the DNA molecule. So that abnormal shape could, or it would be detected by the DNA replication enzymes, and it would immediately remove and replace that base with the right one. So because of the specific pairing system and having just one strand of DNA, the cell can automatically know the sequence of the other strand. So for example, we say that these strands are complementary. So if we have, let's pick a color. Let's try to get blue. So if we have our 5' end that you see here on top and we have the 3' end, because we know what the 5' end is, it means that we know what the 3' end. So again, A always goes with T, right? So we have T, A, G, C, G, 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 T, G, G, A, T, A, T, G, C, 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 A. Okay, so we know that they're complementary. So whatever the opposite is, we know A is with T and G is with C. So the other prime end would just be the complementary nucleotide to that, or nitrogenous base.